Hello, everybody. Thank you for arranging this much-needed webinar. I have special interest in blood clotting and in the identification of novel circulating inflammatory molecules that may be used in the early identification of various diseases. Recently, my focus has also turned to COVID-19 and lately long COVID. Although my research is focused on adults, perhaps there are a few lessons to be learned for the treatment of children with long COVID, particularly if it is taken into consideration that these children will soon be adults. I would just like to acknowledge that the work that is presented here was done with both clinical and research collaborators. My laboratory and our collaborators from various labs over the world have been identifying inflammatory molecules in circulation that might be causing abnormal clotting. And we have also focused our research endeavors on studying the effects of increased circulating inflammatory molecules and how they interact with cells of the hematological system. We focus in particular on platelets and red blood cells, as well as on the clotting protein fibrinogen. We are also interested in identifying novel inflammatory molecules that might play a role in the persistent symptoms of long COVID. Over the years, we have published numerous papers that show the importance of inflammatory molecules in circulation and their role in abnormal clotting. I am particularly interested in platelet signaling and the role in abnormal clotting. Platelets are small, round cellular structures that are in circulation and play a critical role in healthy blood clotting. However, they can become overstimulated and can drive abnormal blood clotting if there are inflammatory molecules in circulation. This can also happen in the presence of viral infections, where platelets can act as important signaling entities. We are also interested in the clotting protein fibrinogen, that is a soluble molecule in circulation, and during blood clotting turns to an insoluble fibronet. During disease, inflammatory molecules and viral products can bind to soluble fibrinogen in circulation, and this clotting protein may become insoluble by forming small microclots. So last year, together with various specialist physicians, we turned our attention to acute COVID-19 and recently also long COVID. It is now well accepted that in addition to respiratory disease, cardiovascular and clotting complications are key threats in COVID-19. Early in 2020, we published a paper where we critically reviewed and discussed our take on clotting and endothelial, platelet, and red blood cell dysfunction in COVID-19. Now, with this in mind, we studied blood plasma and whole blood of patients that have tested positive for acute COVID-19 before any treatment. In the micrographs on the left, you can see platelets are hyperactivated and clumped with numerous extensive feet-like pseudopodia. They have also shed their content and extensive membrane damage can be seen. Note the yellow arrows. In the micrographs on the right, platelet red blood cell interactions can be seen. Here you can also see the spontaneously formed plasma microclots in acute COVID-19. Note the yellow arrows. Now, with this in mind, we turned our attention to long COVID. I will briefly discuss recent findings we have made with regards to lingering hematological and coagulation system failure in long COVID patients. We have noted significant platelet hyperactivation and clumping in the blood of individuals with long COVID. For researchers to study the protein molecule content of blood plasma, we can also use a method called proteomics. Now, without going into too much technical detail, the sample needs to be digested. Otherwise, the sample will cause damage to the equipment. For this digestion step, we used trypsin, a powerful enzyme. Before doing proteomics, however, we first took the fraction of plasma after a first digestion step, and we looked at it after the addition of theoflavin T to see if we could find microclots. We found that the plasma of patients with type 2 diabetes and plasma from healthy patients digested fully after a first trypsinization step. In contrast, microclasts in plasma from patients with both acute COVID-19 and those with long COVID did not digest fully with trypsinization. After a second trypsinization digestion step, we could digest the microclots in plasma from both acute COVID and long COVID patients. We detected various inflammatory molecules that were substantially increased inside the now digested microclots of patients of acute COVID-19 and long COVID versus the equivalent volume of fully digested fluid of the control samples. Of particular interest was a substantial increase in alpha-2 antiplasmin and the various fibrinogen chains that were trapped inside the solubilized fibrinolytic resistant microclots. This molecule prevents clots to lyse, and it is present in plasma of long COVID patients. It might be the cause of persistent microclots in circulations that do not break down via the normal lytic process. We have recently launched a long COVID registry in South Africa. We also plan to study blood samples from a large cohort of patients who are already registered. In this presentation, I wished to focus the attention on circulating inflammatory molecules. Although the inflammatory molecules that affect children with long COVID may not be the same as is found in adults, there is an urgent need to determine which inflammatory molecules are dysregulated in children. In addition, we simply do not know if there are also microclots in circulation of children. Unfortunately, there are no currently easily available pathology tests to look at microclots in circulation, as the techniques mentioned here are all done in a specialized microscopy laboratory. I wish to leave you with a bit of good news and report that at the latest long COVID WHO panel meeting that was held 15 June 2021, clot burden was added as one of the long COVID focuses of the WHO. Thank you for your kind attention.